Bioinfocourse Dr. Öznur Taştan Machine Learning for Molecules and Diseases Dr. Öznur Taştan, 2004 yılında Sabancı Üniversitesi Biyoloji Bilimleri ve Biyomühendislik bölümünden mezun olmuş, doktorasını ise Carnegie Mellon Üniversitesi'nde bilgisayar bilimi üzerine tamamlamış ve sonrasında Microsoft Araştırma Laboratuvarı bünyesinde doktora sonrası araştırmacı olarak çalışmıştır. 2018 Kasım'dan bu yana Sabancı Üniversitesi, bilgisayar bilim ve mühendisliği ile moleküler biyoloji, genetik ve biyomühendislik programlarında öğretim üyesi olarak görev yapan Dr. Taştan, yüksek lisans seviyesinde makine öğrenmesine giriş, lisans seviyesinde veri bilimi ve bioinformatik derslerini vermektedir. Bundan önce Bilkent Bilgisayar Mühendisliği bölümünde öğretim üyesi olarak çalışmıştır. Dr. Taştan'ın araştırma konuları işlemsel biyoloji ve makine öğrenmesi alanlarını kapsamaktadır. 2012'de L'Oreal UNESCO Genç Bilim Kadını Ödülü'ne, 2014 yılında ise Bilim Akademisi Genç Bilim İnsanları Ödülü'ne, 2019'da ise Mustafa Parlar Araştırma Teşvik Ödülü'ne layık görülmüştür.
Sayın Dr. Öznur Taştan ve değerli katılımcılarımız, Bioinfocourse kapsamında gerçekleştireceğimiz etkinliğimize hepiniz hoş geldiniz. Bugün hocamız bizlere Machine Learning for Molecules and Diseases konulu bir seminer verecektir. Hocam tekrardan hoş geldiniz.
Çok teşekkür ederim. Hoş bulduk.
Dersin sununuza başlayabilirsiniz.
Teşekkür ederim. Bu güzel davet için teşekkür ederim. Öğrenciler çağırdığı zaman pek kırmak istemiyorum. Biraz geciktirdim bu konuşmayı. Doğum izninden yeni çıktım. O yüzden kusuruma bakmayın. Bu arada yani bırakın sunum yapmayı, konuşmayı bile unutmuş gibi hissediyorum. O yüzden sürçülisan edersem affola. Ben slaytları İngilizce hazırlamıştım. O yüzden İngilizce sunacağım çünkü o bir türlü böyle ortaya karışık bir şey oluyor. Ama yani soruları Türkçe de sorabiliriz. Her şekilde devam edebiliriz. So, thank you for the kind invitation and the introduction. So the title of my talk is the Machine Learning for Molecules and Diseases. So I know that this is a very big topic. I kind of cheated. I made this big heading so that I could choose at the very last minute what I'm going to tell you. So as you know, machine learning is everywhere. I mean, it's transforming our technologies, everyday technologies that we use. Like you might have seen the big advances especially in the vision problems. Like this is a live object recognition system, or in the voice recognition systems, or in the translation systems, or like in this one, you are actually creating a website automatically by describing it with a text. In agricultural, in business, in different domains of the business and industry, and we are hoping to have like self-driving cars so that we don't have the burden of driving ourselves. And even in it's pushing the boundaries of the disciplines. So it's helping many disciplines, including astrophysics, like the black hole image that we had was actually made possible also by using machine learning models, physics-informed machine learning models. So these advances were fueled by the data, right? And we have the term of astronomical data, but actually the biological data we have is much more larger than the astronomical data. So we have this very nice title of a paper that came up in 2015. These are estimates from the 2015 compared to the amount of data that's produced on Twitter, YouTube, which we could guess that a lot, right? A petabyte is like 10 to the 15 bytes. And genomics data alone is like, it's going to be many folds by 2025. That's the estimate. We don't only have genomics data, as you might know as students who are interested in bioinformatics, that in biology we have all types of data. it's rich both in size and also in structure as well. So we have omics data, genomics data, epigenomics data, transcriptomic data. We have like recently microbiome data, right? And we have also like structural data that has been deposited from the tedious experiments like X-rays and NMRs, like protein structures, RNA structures, drug structures, other molecule structures. We have functional data that's coming out of like large-scale, small-scale experiments that are annotating genes, annotating other molecules in the cell. We have also cell types coming with the single-cell sequencing, atlases of cell types, and we have also network data as well. Like this network data come in different forms. Like we have like pathway data, we have protein-protein interaction data, all regulatory networks data, so all kinds of network data. So not surprisingly, machine learning has been applied to this domain pretty early on. So this is not kind of a new advance like, okay, machine learning is hype and let's solve problems in machine learning, in biology with machine learning. But since like many decades, people have been using machine learning to solve these problems. What is maybe interesting for this time is that the amount of this data is large and it's easier to access, and we have better machine learning tools. So, but why do we want to solve these problems with machine learning? Like, does every computational problem that we have require machine learning? No, right? I mean, so learning typically outside of the biological domain as well is useful when humans are unable to explain their expertise. For example, when your mother calls you, you know that it's your mom, immediately you do a speech recognition. But we don't. If I were to write to you, what is the formula that your brain used to do that speech recognition, you wouldn't know that, right? You are very good at it, but you don't know how it works. It's similarly in our cells. Lots of computations are being done, right? The molecules are recognizing each other in very specific manners, building structures, macromolecules and stuff, fighting with the viruses and the bacteria, but we don't know how we are doing this, right? So it's a very complex system that we cannot explain how the expertise of our cells and tissues and organs. Machine learning is also useful when you need to adapt solutions to particular cases. So like, I think Amazon, right? They have millions of customers. They cannot write another program for every customer to recommend what items to buy. So their solution needs to be adapted to particular cases. Similarly with the Google. So Google search is adapted to different users, even Google Inbox and so on. So many of these tools are personalized with the data that we have in biology too. So we have like patient data, right? So we want personalized medicine to achieve like the best medicine. So solution needs to be adapted to people or particular cases. And a third domain where machine learning is useful is like when the human expertise doesn't exist at all. For example, when we have robots that are sent to Mars, no human know how to navigate on Mars, right? So the robot learns itself by the interaction with the environment. In biology too, there's lots of problems, lots of questions and problems that we don't know how to solve them, but we have the data that could actually inform us. So in our lab, we do this kind of problem. So we are fascinated by the biology and the impact that the solution could bring to patient lives and the basic science, and we are also fascinated by the machine learning algorithms and like the math and the computing background behind them. So starting my PhD, I have been working on this intersection, so application of machine learning in biology. But this application is not like you take a oftentimes what we need to do is we need to kind of be creative and define the biological problem well, so that it actually fits the machine learning framework well, and do not fall into the pitfalls of the machine learning when doing that, and also come up with some creative solution to be able to solve the problem, the biological problem at hand. Oftentimes, taking the off-shelf method and like applying it to the problem fails miserably. So what I'm going to do is like to kind of demonstrate what we do in lab. It will teach you by example. I will show you 2 example projects which are kind of very different from each other. Like one of them uses patient data, the other one uses, like the other one is about RNAs. The first one uses omics data, the second one uses sequential data. This also kind of on purpose to show you that like we have lots of interesting problems and also interesting data types in biology. So let's start. I might have to kind of skip some of the details of these projects, and if you have questions, feel free to write to me afterwards or ask me questions at the end of the talk. So I'll start with the cancer patient subgroup identification. So this was a project funded by TÜBİTAK as my career project. So these are students who kind of worked. Some of them were the leaders of the project. Yasin, Ali Burak, and Beyza are mainly the main contributors, and Ali Osman and Furkan helped also all the project. So this is also one part of this project is published in Bioinformatics in 2020. So there's a paper if you need to see the details. So what is the question? So in traditional medicine, what we have like one drug for the population, right? And there could be subpopulations inside this population. They will have an adverse effect. Some of them would have no benefit. Some of the patients would benefit from that. If we were to know these subgroups beforehand, and then we could maybe design better therapies. But I mean, the ultimate aim is the precision medicine. That's like using the genomics and the omics data of the patient, their lifestyle, all these medical records, to be able to design a therapy or a drug for per person. But that's kind of the long distance goal. In between these two, there's something called stratified medicine, which is like identify these patient subgroups and then try to develop therapy for these patient subgroups. So it's critical to know what these subgroups are beforehand. And many complex diseases are like that. You have patients that are not that have subgroups of diseases within itself. So cancer is the typical case. Like in cancer, you have subgroups very frequently, and it actually hinders development of like better diagnostic tools, this heterogeneity. Therefore, if you were to find these subgroups ahead, you can actually design per group a diagnostic tool. You can also get molecular insights on the molecular mechanism of the subtypes or each of the subtypes, and you can maybe come up with better treatment strategies for each of the subgroups. So it is important to be able to know what these subgroups of patients that are more similar to each other. That's the problem. That's our goal, how we are going to do that. So one advance was like the omics characterization of the cancer patients, like spearheaded by the projects like the Cancer Genome Atlas project, where cohorts of patients are sequenced for different types of data. So this having like different types of data on the same patient is kind of similar to, so the finding subgroups of patients is a clustering problem, right? Now we have like multiple data on a patient. This is actually a familiar problem from machine learning, which is called multi-view clustering. So in this multi-view clustering, you have kind of a view of the data. You have different views of the truth, and then you try to combine them. In this case, we have like different reports of what is going on in the tumor cells, and we try to combine them to find the better clusterings. But this is something that people have been studying since 2009, maybe even earlier, and there are many methods which ranges from early integration, where you concatenate the features of coming from different omics data and then cluster, or you do some intermediary step and find clusters, or you do late integration, find clusters from each of the data set and then combine them by looking at their consensus or agreement. So what we are going to do is something, an intermediate integration, like they will take these data sets, then we will crunch some numbers and we will try to find the clusters. So this is one thing, like we are going to use multi-view clustering. The second thing is like, one challenge is like the tumors are quite heterogeneous. So when you look at the patients, what you would see is like, it's very rare that you would see common mutations across patient sets. Like this graph shows you the frequency of the mutations on for the genes. So there are few genes which are mutated in many patients, but most of the genes are only mutated or altered, I would say, in few patients. So, but if you were to put the biological context in place, it could be different, right? For example, this patient 2 and patient N, they don't have exactly identical alterations. So imagine these are like, this is a pathway turned into a graph, and these nodes are genes, and genes, the color indicates whether this is mutated or not, right? Patient 2 and patient N have only 10 as identical. 5 and 4 is different. 9 and 8 are different. But 5 and 4 are neighbors. 9 and 8 are neighbors as well. So if I were to compute the similarity across these patients, I should be able to say that patient 2 and patient N are similar, not only because they share the mutation in 10, but they also share mutations that are neighbors of each other, right? So we want to take into account the molecular interactions that we know from the literature. And this idea is also not that novel. I mean, people have been doing that. What we did was like to combine these two ideas in a kind of novel way. So to do that, we use the kernel functions. If you have used a support vector machine, you might have used radial basis function kernel, for example, or a polynomial kernel. So the kernels are actually, they will take two objects, and then by computing an inner product in a Hilbert space, they would measure the similarity of these two objects. So it will take two objects and return you a value, and that value will indicate how similar these two objects are. So if we were to do a computation in our original data, we would come up with features. For example, this could be genes, and one feature could be like whether this gene is mutated or not in that patient, right? So we will have a patient by feature matrix, and I use this. But when we work with the kernels, what we do is we compute a similarity, kind of a kernel matrix, patient by patient, and each of the cell will be the evaluation of the kernel function on these pairs of patients. So if this was a radial basis function kernel, for example, you would put the feature vectors of these patients and then compute the difference, and then e to the minus blah blah, the formula, and then you will get one number and put that there, right? So it's like pairwise comparisons computed via the kernel functions. But what we have are, if we want to take the molecular context into the account, again, imagine this is a pathway turned into a graph. We want to compare graphs. So we want our objects are not simple feature vectors, but graphs. But these graphs are also node labeled. So node labeled based on the patients. As you can see, the graphs are identical, so their topologies are identical, but the labels of the nodes will change based on the patient alterations. So, and of course, assessing the similarity of graphs has been also studied in the literature, and this is studied under the name graph kernels. So the graph kernels will take two graphs and then assess the similarity between these two graphs. And there is quite a lot of literature. If you are interested, you can look at this encyclopedic guide review paper. It does a very good job in organizing the properties of the kernel. So we have lots of actually graph kernels that has been proposed. So do we need another one? So one thing about these graph kernels are that they are actually trying to solve a more difficult problem. They want to know whether two graphs are similar in terms of their structure. In our case, the structures are going to be the same. They are going to come from the pathways in this case, but we extended this work a little bit further to protein interactions and 3D genomes. But let's say the pathways and the graphs. So the graphs are going to be the same, but the node labels of the graphs will going to be different. So if you were to, and this is a patient by patient kernel matrix, and if the color is if the similarity is 1, then you will see a color of black. So if we were to compute these kernels, the well-known, very good graph kernels, propagation kernel, Weisfelder, Lehmann, or Graph-Hopper, most of the matrix will be black. And this is because it will evaluate the patients very similar to each other because the graph structures are the same, right? The node labels will only contribute a little. So what we want is we want to take the graph structure into account, but what we want is actually the difference in the similarity in the node label distribution. So we designed a new kernel called Smoothed Shortest Path Graph kernel. The idea is you have patient graphs, which, let's say this is an alteration of in a patient, and you smooth those labels on the graph. That means that the neighbors will also receive some alteration. So the G hasn't been altered, but because its neighbor is altered, it will change as well. And we just show a small snippet of the graph, but imagine this is big graph, so different neighbors will also send their alteration flows to these ones. And this is actually also studied a lot under name network propagation. So you can do that using random walk. And after you smooth away, that means that you bring the context now into account, the network structure, and you compute these smoothed feature vectors across the patients. And to do that, we find all pairs of shortest paths on the graph and then compare their node label distribution. And if that number is high, that means that these patients are going to be similar to each other. Okay, so combining idea 1, idea 2, we propose something called PAMOK. This is as follows. So we have our different data sets like mutations, protein expression, gene expression. This could be extended, but in this case, we only use these 3 types of data. And per patient, we have this information. And we have graphs coming from the pathways. These pathways are like on average like 100 nodes, but here to show they are small. And we have like M pathways. We map these alterations on the pathways for each patient. So for patient 1, I have M pathway that are labeled. For patient 2, I have M pathway. And then I'm going to compare on the same pathway the patients and compute a kernel matrix per pathway. So in between, I'm doing a graph kernel computation pairwise for all patients and getting a kernel matrix for each pathway. At the end of the day, I have several views of how similar these patients are, right? And then the next step is to apply a multi-view kernel clustering method to get the patient clusters. So let me skip these ones. There are actually quite a lot of multi-view kernel clustering methods to use, and we have actually systematically analyzed like which one we should be using. Here, the y-axis, if it is large, it's good. This means the clusters are well separated in terms of their survival rate. So this is actually the evaluation metric that's used in this work. So if you arrive at clusters and then you look at the survival distribution of these clusters and then conduct a log rank test, which tells you if they are very well separated in terms of their survival, the p-value of the test will be low, so the minus log 10 will be high. So which is something good. We want like clusters that are different in terms of their survival distribution, which is an indication that this could be useful in the clinic. So we use different methods and compare them and come up with concluded that this one works good for us, multiple kernel k-means with matrix induced regularization method. And next, we also evaluate the graph kernels, like how well this graph kernel that we propose work compared to the other ones that exist. So this is radial basis function, this is propagation kernel, and so on. And then we see that it does well. These are actually done on the kidney cancer curve. And to evaluate it further, we did actually a bootstrapping. So we did this experiment. We sampled the patient set several times and then redo the clustering and then assess how well the clustering is using the p-values. And you get a distribution per method. And then we compare them. So the higher this box, then it is better. So this is the proposed method. Some of these methods are also published like in Nature Methods and so on. So we did pretty well in this cancer type. So this at the end, when we apply the method to the patient group to find the patient groups, this is the Kaplan-Meier curve that we get. Some of you might be familiar with this. What this shows you is like each color is a different cluster, and this is days, and this is the survival probability. So as the time goes, the survival probability will decrease for the patient group. And for example, for this group, it decreases pretty fast. The separation between these lines is the important one, and when you assess this with a statistical test, what you get is e to the minus 11, which is very good. And we also check whether this is due to the age or gender. We find out that it's not. We also check whether other clinical parameters are different for this patients group. They were, which is and also an indicator that this could be an interesting grouping and so on. So this shows the distribution of these parameters across groups. One nice thing about this method is like at the end of the multi-view clustering, the multi-view kernel clustering method that we use will assign weights to each of these kernels that we computed from the pathways. These weights are an indicator of which of the pathway were more informative in computing these clusters. So we can extract them and then look at like kind of the importance of the pathways. And when we do that, when we sort these pathways, we find P53 effectors, which is kind of no surprise for all cancers, this comes up. And then, but the other ones, this one is also very common for cancers. These endothelins and especially HIF1 alpha transcription factor network is very specific to KERK. So interesting pathways comes up. So to sum the first part, PAMOK evaluates patient similarities based on their molecular alterations in the context of known pathways using a novel graph kernel. And this also allows you to find pathways that are of importance. We extended this work. Beza, whose picture I showed you at the beginning, has extended this work to work with protein interaction networks, where she designed a new kernel that computes the similarity based on the communities and their communities on the protein interaction network. And also, she also augmented that network with the 3D chromatin interaction data. So when we applied PAMOK across different cancers, it doesn't always work best. Some of her new contributions also work best for other cancer types. But I didn't include those here yet. So the second, it's already half an hour, so this part will be a little bit quicker. So in the second part, I'm going to tell you another project from another master student, which is very different than cancer. So this is about non-coding RNAs, particularly long non-coding RNAs. So if you look at the human genome, only a very small fraction of our RNAs are actually translated, which are like the messenger RNAs, right? So, and the rest is kind of all kinds of non-coding RNAs. We have small non-coding RNAs, we have long non-coding RNAs, like different kinds, and they are actually their functions are being discovered. They are less studied, but they have various role in transcriptional regulation and so on. But the categorization of the long non-coding RNAs is a little bit arbitrary. What we call long non-coding RNAs are those that are longer than 200 nucleotides. And so it is by definition non-coding, right? But when you look at the literature, many reports arrive that they say that, for example, a micropeptide encoded by a putative long non-coding RNA regulates muscle performance. So a peptide encoded by a putative link RNA. So there are several examples of this. So it's been reported that some long non-coding RNAs might contain regions which code for small micropeptides, small proteins. And these open reading frames are the regions which are translated, right, are missed, maybe because of this arbitrary definition of that. The traditional criteria of what makes an open reading frame is like it has to be 100 amino acids in eukaryotes and 50 amino acids in bacteria. So because of this arbitrary decision, maybe that's why some of them hasn't been studied and missed. So there are actual ways to capture these the translated RNAs in the cell. And for example, ribosome profiling is one method. What you do is you look at what transcript the ribosome is working on, and then get the sequence and then map it to the genome and figure out whether it is translated. But this is cell type dependent, it's expensive, and there is also mass spectrometry, which is not very sensitive. So there are experimental methods, but they are not perfect. So we thought maybe we could find a methodology to assist these things. So Afshan actually worked on this project. Berke helped, and this is also in collaboration with Ogün Adebolu at Sabancı University. So the aim is like, let's develop a computational approach for discovering which long non-coding RNAs that might be coding. So this work is under revision right now. We need to work on the revision. So there is actually a lot to be done maybe in this project. So here what we did. So we got coding sequences and non-coding sequences. So, and then this is our input, and then you get sliding windows of trimers. So you represent them in a way that is that can be inputted to a machine learning model, right? So you get the features of a numerical representation of these DNA sequences of that codes for the coding RNAs or the non-coding RNAs. And then we learn deep sequential models. So we tried several architectures, so 3 of them that does well. One of them contains the CNN, the other one includes an STM, the other one includes a transformer. So these are like popular architectures that are used in natural language processing to process text language, right? So here we are using it to process the protein DNA language. So the idea is that let's train these models, and then let's look at the training dynamics of these models. What do I mean by that? Let's look at, like, if, for example, if there is a, maybe let me first tell you how well these models do, and then come to that. So these different models actually are pretty good. So they can discriminate the coding versus non-coding pretty well with a F1 score of 0.94. So they work well. And now what we did was, so here this graph is maybe more helpful. So when these are different epochs of the training. So if you have a non-coding sequence, let's say this B, and here on the y-axis you have the estimated coding probability. So the model is, although this is a non-coding sequence, the model is saying that I predict this is a coding sequence. I predict this is a coding sequence. And across the epochs, it's kind of consistent. Like, it's like here, if you look at this RNA2, its true label is non-coding, but the prediction probability is it's coding, right? So it could be a mistake. The model might not have been able to learn that kind of sequences, but it could also be the case that this is actually misannotated. Maybe this actually contains some coding region that leads the model to think that this is a coding sequence, right? So what we do is, like, when we train the models, we save the predictions for each epoch and then analyze these curves. So we calculate the mean of the predicted true label probability, and then the standard deviation of that probability. And if consistently the model is saying that this is a coding sequence, although it is labeled as non-coding sequence with a small standard deviation, then we filter them as candidate misannotated link RNAs. So we do that for 3 of the models, and then combine the set. So there is quite an agreement between them. Some models are more conservative to call, some are not, but there is some region that is overlapped by most of them. And we compare these candidate non-coding RNAs also with other experimental evidence. So these are other methods or techniques or databases that report some coding non-coding RNAs that could have a potential to be coding. And we see a large overlap with these ones. So what we did next was to, we couldn't do this for all the candidates that we have, but we did this aggregating other evidence part for all the ones. Like, we blasted these sequences and see if there is a similarity of this non-coding sequence to any coding sequence, right? That could give an indication that this could also be coding. We looked at mass spectrometric data that is available, ribosec data, and other coding potential tools. And for a subset of them, we also run AlphaFold predictions. So here I show you one of the interesting results. So this is like, in the database, this is a long non-coding RNA, but inside this, we look for all the operating frames and then blast them. There is actually a blast hit that is similar to this region. These other tools also say that it could be a coding one. And then we looked at for the CPAT and CPC2, actually, they say these are other coding potential prediction tools. They say the coding probability is not that large, except CPC2 maybe. But in our models, the coding probability is quite large for this one. And when we run the AlphaFold 2 prediction, it actually nicely falls into a helical structure. There is also evidence in the literature that most micropeptides, not related, not only to non-coding, but in general, micropeptides falls into a helical structure. So we are now at the stage of like analyze this list in different ways, maybe looking at the conservation of the sequences and like doing predictions for all the sequences and so on. So in conclusion for this part, we suggest a way to come up with sequences that could come up with long non-coding RNAs that could actually be misannotated as non-coding, but contain some regions that can code for micropeptides. And this actually offers a promising strategy to assist the experimental efforts in characterizing the hidden proteome of the encoded in these long non-coding RNAs. So in the remaining maybe 5 minutes, I'm gonna go over with some other projects that's been recently that we have been recently working in my group. Not all of them, but some of them. So we have been looking at like how to predict post-translational modifications. Like Berke is working on these post-translational modifications are like once the protein is folded, then various chemical changes could be happening on the protein, and this actually increased the repertoire of the protein structure because the protein will be in a state with the post-translational modification and in another state without the post-translational modification. So some functions depends on the presence or the absence of these modifications. So one of these modifications is simulation, and Berke has been working on like how to, when you are given a protein sequence, can you predict whether the site is simulated or not using some natural language processing techniques. Before we have worked with Iman, a very talented student who is now doing a PhD in US, on kinase phosphatide association predictions, but for understudied kinases, which require us to go beyond the classical supervised learning approaches, where you need abundance of data for the kinases. In this case, we use a zero-shot learning model, which is a specific technique to deal with this kind of data. In collaboration with the Ercüment Çiçek lab, we also work in several drug-related projects. Like in this one, Halil works on this is published now on predicting synergistic drugs. So given 2 drugs and the cell line and gene expression data can predict whether these 2 drugs will be working synergistically or not, and compared to the state-of-the-art methods, we do pretty well in this task. In another project, again with the Çiçek lab, we worked on predicting drug side effects. In this case, the data is the drug structure. We also use the gene expression data, and the labels are the different side effects that this drug can have. It's a multi-label prediction task. This is in collaboration with the Sündüz Keleş lab from University of Wisconsin Medicine. Here we have, this single cell data. Imagine you obtain the single cell expression data for different subjects and for different time points. And to be able to, and we want to discover the gene clusters at each time point per subject, right? We would assume that the across time, the changes will not be that abrupt, and across subjects, the changes are not that abrupt. And using these constraints, we developed a spectral clustering method to find these communities. This is also a work under development, I would say. Again, this is Ali's main master's project, which is quite different than what we typically use, study not molecules or diseases, but eventually will evolve to there. This is Drosophila sleeping, the flies sleeping in chambers, and then we have the videos of these flies, and we try to find the different movements from these different movements of the flies across during the night, different sleep states. And Ali has done a lot of work on trying to figure that out, like to annotate what type of activities they do and so on. And eventually we will find, for example, how different phenotypes are shaped by the genes and so on. But this is kind of long distance goal. With Ogunadebale lab, we also work on the variant effect prediction. This just recently came up last week, this work, using phylogenetic trees. How can we predict the variance effects in a better way? This work works on microRNA cooperation. So microRNAs binds to mRNAs and regulate their expressions, and which of the microRNAs will be synergistically working. So this was Gülden's PhD thesis. Now she's a postdoc at NCI. And we also started working on electronic health records, which is like digital patient records on the hospitals, and try to predict patient-related things, for example, in ICU mortality and so on, using the rich data that is recorded on the patients of the patient during the hospital stays and so on. So these are kind of two sample. I mean, there is not enough time to go all of them, and it will be boring, I think, to listen me for hours. But in conclusion, I mean, biology is rich, right? So it's like rich with data, it's rich with problems, and many of them are amenable to machine learning. And the nice thing about it is like the solutions can advance both basic science, right? We can learn more about how these molecules are working, and it could also advance the medicine and like improve patient care. So this is kind of our ultimate goal. I don't know whether we will see that impact immediately, but in the long run, hopefully. So I would like to thank to all members and collaborators of the Taştan Lab and the funders that support our work. And I would be happy to take your questions. You can ask in Turkish and English, whichever way you want. Thank you.
Değerli sunumunuzu verdiğiniz güzel bilgiler için çok teşekkür ederiz hocam.
Rica ederim.
Dilerseniz ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru cevap kısmına geçebiliriz.
Tamam. Ben de şeyden çıkayım. Share screen'den.
İlk sorumuz, kategorik dizilerde yüksek boyutsallık ve heterojenite sebebiyle veri analizlerinde çeşitli zorluklar yaşandığı bilinmektedir. Bu konuda çözüm sunacak çalışmaların daha işlevsel olması için ne gibi özelliklere sahip olması gerekmektedir?
Mesela kategorik ve heterogeneous deyince aklıma mesela bu son gösterdiğim tezde, Ege'nin tezinde elektronik health record'larla çalışıyoruz. Burada bolca heterogeneous ve kategorik veriler var. Mesela hasta, değil mi? Hastalık, hastanın teşhis olduğu hastalıklar. Hastanın geçmişe bir sürü binlerce hastalık var ama hastaya baktığımız zaman aslında geçmişinde çok belki en fazla 10 tane hastalık geçirmiştir. Böyle bunu biz mesela bir one hot encoded denilen 0-1 vektörüyle göstermeye kalksak, kocaman bir vektör içerisinde de çok az 0-1'lerin olan sparse bir vektör olur ve bu gerçekten sıkıntı, öğrenirken zorluklar oluşturacak machine learning algoritmasına. Genelde istediğimiz şey hani embedding, bunu böyle daha dense ama anlamlı vektörlere embed edebilmek. Onun için de mesela o çalışmada biz işte knowledge graph embedding yapıyoruz. İşte bütün bu veriyi bir knowledge graph'ta gösteriyoruz. Sonra o nodları machine learning, knowledge graph embeddingle daha dense bir vektörle represent ediyoruz vesaire. Biraz tabii hani probleme ve veriye ve ne kadar heterojenite olduğuna, ne kadar sparse olduğuna göre değişen bir şey ama farklı teknikler var, evet.
Cevabınız için çok teşekkür ederiz hocam. Diğer soruya geçiyorum.
Görebiliyor mu olmam lazım?
Şu an ekranda aşağıda yansıtılıyor. Yani sorular okunduğunda aşağıda yansıtılıyor sorular.
Tamam. Tamam, geldi.
Nearly all machine learning algorithms, particularly deep learning approaches, are still under investigation on how the model arrived at its final predictions. Even though various explainable or interpretable methodologies have recently been proposed, what could be the reason why the machine learning-based model cannot provide enhanced explanations for the reliability and interpretation of the models?
Bunu İngilizce mi cevap, Türkçe mi cevap? Türkçe cevap diyeyim. Şimdi soru çok
Evet.
güzel bir soru. Evet, bu modellerin explainable ve interpretable, yani tarzanca oldu. Interpretable olmasını istiyoruz. Çünkü özellikle biyolojide ya da hasta verisinde bu model bunu niye predict etti diye bilmemiz lazım. Yani hani doktora gittim, benim prediction 0.85, hastayı kes diyemeyiz. O yüzden hani bu model niye bunu böyle predict etti açıklamamız lazım. Biyolojide de aynı şekilde hani buyur benim prediction'ım 0.95 ile bunu böyle predict ediyor. Lütfen işte 100.000 liralık şey alıp saf malzemesi alıp bunları test eder misin diyemiyorsunuz ve açıklamanız gerekiyor. Bunlar ama yani siz de bu soruyu sorduğunuza göre takip de ediyorsunuzdur. Research konuları, yani şu an limitli, hala açıklayabileceğimiz şeyler limited, ama bunun üzerine yapılan çok güzel araştırmalar da var ve bazı yollar kat ediliyor. Her şeyi açıklayabilecek miyiz? Ondan emin değilim. Yani hani deep learning'in mesela çok karmaşık bir fonksiyonla çıkarttığı bir şeyi açıklayabilir ve gerçekten interpretable olacak mı bilmiyorum. Ama belki başka yollar deneyeceğiz. Hani bunu şöyle değil de böyle olsaydı bu feature sonuç ne olurdu? Böyle olsa, yani perturbation based şeyler olacak belki. Yani bununla ilgili bir sürü şeyler çalışıyor. İşte sharp value'lar var. Lime diye bir metot var. Böyle işte biliyorsunuzdur. GradCam metotlarını Vision'da çalışılan. Attention, nereye attend etmiş model gibi şeyler var. Günün sonunda hani bir sürü kişi çalışınca güzel bir şeyler çıkıyor bilimde. O yüzden umudu kaybetmemek lazım bence.
Hocam, cevabınız için çok teşekkür ederiz. Bir diğer sorumuz katılımcımızdan geliyor. Hocam, normalizasyonu yapılmayan ve kararlı yapı göstermeyen veri kümelerinde ne tür makine öğrenmesi modelleri kullanılmaktadır?
Kararlı yapı göstermeyenden kasıt ne? Tam onu anlamadım. Onu açıklarsanız yaparım. Normalizasyonu yapılamayan, yani tree-based modeller aslında illa normalize etmenize gerek yok. Yani tree-based, yani genelde biz başka modellerle karşılaştırma yaptığımız için datayı normalize edip işte onlarla işte karşılaştıralım diye işte ne bileyim gradient descent normalizasyon yapılmazsa etkilenir. Ya da logistic regression vesaire gibi şeyler bunlardan etkilenir. O yüzden normalize edip de çalıştırıyoruz. İşte SVM etkilenir. Ama aslında decision tree'ler tek başlarına normalizasyondan etkilenmez. Yani normalize ettim bu çıktı, normalize etmedim farklı çıktı değil. O yüzden hani öyle bir veri tipi varsa öyle tree-based model'lar olabilir.
Cevabınız için çok teşekkür ediyoruz hocam. Diğer soruyu okuyorum. Güvenilir ve analizi yapılabilir, kaliteli veri üretmenin zor olduğu hastalıkları ve molekül yapılarını incelemek için data üretiminde machine learning kullanılmakta mıdır? Örnekler verebilir misiniz?
Güvenilir ve analizi yapılabilir, kaliteli veri üretmenin zor olduğu hastalıkları ve molekül yapılarını incelemek için analizinde kullanılıyor. Genel, yani soru herhalde doğru anladıysam şimdi bir data üretimine gelince mesela data işte data augmentation falan gibi yöntemler kullanılıyor. Bir de generative advice GAN'lar gibi, mesela işte bu drug molecule structure yapalım, üretelim. Şu target'a bağlanan yeni bir drug üretecek, compound üretecek olsak ne üretirdik? Onun için generative adversarial network gibi şeyler kullanılıyor. Ama bu soru galiba başka bir şey soruyor. Yani elimizde data olmadığı zaman ML kullanabilir miyiz? Ben bazen işte mesela bizim bu zero shot learning kullandığımız problemde predict etmek istediğimiz kinazlar vardı ama kinazların training datası yoktu. Çünkü çok az çalışılmış kinazlardı. Orada zero shot learning kullandık. O da bir nevi şöyle bir metot. Yani bilinen kinazlar üzerindeki datayla eğitiyorsunuz. Sonra da kinazlar arasındaki benzerliklerden yararlanarak bir nevi transfer ediyorsunuz bilgileri. Yani böyle birazcık daha komplike şeylerle bence olabilir. Yani
Tamam
burada da
hocam.
mesela o hastalığın başka hastalıklara benzerlikleri ya da o molekülün başka moleküllere benzerlikleri yararlanabilir.
Tamamdır hocam. Çok teşekkür ediyorum. Bir diğer soruya geçiyorum. İlaç geliştirme aşamaları arasında gelişmiş iletişim ve etkili olmayan moleküllerin klinik gelişimin sonraki aşamalarını geçmesini önlemek için düzeltici eylemlerin gerçek zamanlı olarak gerçekleştirebildiği öz yinelemeli bir yaklaşım, farmasötik endüstrisinde modelleme olarak kullanılabilir mi?
Sorunun başını tekrar görebilir miyim? İlaç geliştirme aşamaları
Tabii ki de.
arasında gelişmiş iletişim ve etkili olmayan moleküllerin klinik geliştirme geçmesini önlemek için. Ya bu şimdi bu drug işi makine önceden hani kimyacılar, işte keminformatikçiler çok çalışıyorlardı. Bayağı da üzerine çalışılan bir konuydu. Ama şimdi machine learningçilerin daldığı bir alan oldu ve hani bir sürü makale geliyor. Özellikle işte graph machine learning çalışan expertlerin hemen hemen hepsi işte drug'ı nasıl elerim sistemden, işte nasıl yeni drug molekülü generate ederim, drug'ın molekül özelliklerini nasıl predict ederim vesaire gibi birçok çalışma var ve yapılıyor yani işte mesela bu RNN'ler vesaire bu smile sequence'ları denilen mesela structure'ı bir sequence olarak represent ediyorsunuz. O sequence'da işte parantezler var, harfler var vesaire ama sequence olarak represent ettikten sonra sequential modellere input edilebiliyor işte. RNN'lere girebiliyor mesela orada yenilemeli modelden kasıt o sanırım. Öz yenilemeli modelden kasıt. Yani ondan sonra CNN'lere, RNN'lere, LSTM'lere sokabiliyorsunuz modeli. Yapılıyor bayağı yani her şey yapılıyor. Bazen böyle hani bir fikir geliyor, süper fikir diyorsun, Google'lıyorsunuz, yapılmış. Ama bu çok yıldırmasın çünkü yani her paper'ın bir improve edilecek tarafı oluyor vesaire.
Cevabınız için çok teşekkür ediyorum hocam. Bir diğer sorumuz katılımcılarımızdan geliyor. Bulut ortamında veri toplama ve makine öğrenimini görevlerinin hesaplanması, hassas verilerin sızması konusunda endişeleri neden olmaktadır.
Hı hı.
Çok sayıda analiz, deney ve değerlendirme yoluyla homomorfik şifrelemenin doğruluğu etkilemeden makine öğrenimine uygulanması mümkün müdür?
Mümkün, evet. Bu da çalışılan bir konu. Bizim Sabancı Üniversitesi'nde de çalışan hocalarımız var. Berkay Savaş özellikle. Şimdi homomorphic AGI, yani bununla ilgili Microsoft'un falan da var çalışmaları. Bu tam aslında hani bu işlerin uygulanacağı domain. Yani hasta verisi var. Hasta verisinin dışarıya sızmaması lazım. Hani sadece researcher görsün, başka kimse görmesin. Ben burada nasıl işte secure computation yaparım machine learning'le bu data üzerinde. Bu hot bir topik. Çok çalışılıyor. Hani bunda ilaç şirketleri de çalışıyor. Bir alternatif de işte federated learning falan yapıyorlar. Ama yönü çok açık bir alan. Benim bu arada bu PAMOK'ta çalışan Ali Burak Ünal öğrencilerimden bir master benle yapan, o Almanya'da bu privacy preserving machine learning konusu üzerine doktorasını yaptı. Homomorfik şifreleme üzerine değil ama hani farklı privacy preserving yöntemleriyle, onlarda da sanırım HIV verisi vardı. Bir sürü use case'i var ve hakikaten bu önemli bir alan. Çok işe yarayacak, gerekecek, bütün hastanelerin, araştırma merkezlerinin ihtiyacı olacağı bir şey olacak.
Cevabınız için teşekkür ederim hocam. Bir diğer soruya geçiyorum. Kişiselleştirilmiş tedavilerde ilaç kombinasyonu stratejilerinin deneysel doğrulamaları büyük öneme sahiptir. Bu konuda ilaç kimya yapısı ve hücre içi profillerini karşılaştırmalı analiz sunabilecek modeller bulunmakta mıdır?
Güzel soru. Şimdi bizim bu drug matchmaker dediğim sinerji prediction modelini biz hücre hattı verisiyle eğitmiştik. Yani hücre hattı ne oluyor? Hasta verisi çok eskiden bir hastadan alınmış ama çoğaltılmış. Şimdi bunu şu an halinin üzerine çalıştığı da benim elimde mesela bir hasta geldi. Bu hastaya özel ben bu prediction'ı nasıl yapabilirim? Bu da open bir problem. Yani işte üzerine çalışıyoruz şu anda. Ama dediğiniz gibi yani hücre içi gen expression profilleri önemli. İlacın kimya yapısı önemli. Bunları ama nasıl kullanacağız da modele bu bilgileri vereceğiz ve işte bu hasta için bu çalışır, bu hasta için bu çalışmaz diyecek vesaire. Şu an çalışıyoruz. Başkaları da eminim çalışıyordur. Buradaki sıkıntılar şey oluyor ama hani yaptıktan sonra bunları test etme işi. Bu test işini pek kolay yapamıyoruz burada. Hani dediğiniz gibi deneysel çalışan insanlar lazım ki hemen biz hızlıca bunları bulduk, yapalım, test edelim. Belki oradan gördüğümüz bir şeyle gidip modeli farklı şekilde eğitiriz. Yanlışımızı düzeltiriz vesaire. Olup çok efficient çalışmıyor şu anda bizde. Yani bizim lab için en azından.
Cevabınız için teşekkür ederim. Bir diğer soruya geçiyorum. İlaç endüstrisinde açık veri paylaşımı yaygın değildir. Bu sebeple seyrek verileri işleyebilen bir algoritma geliştirmek sorunu çözümünde ne kadar efektif bir yaklaşım olur?
Seyrek veriden kasıt küçük veri aslında herhalde, değil mi? Evet. Yani zaten küçük veriler için hani classical machine learning algoritmaları, işte bu gradient boosted yöntemleri vesaire gayet iyi çalışan yöntemler ve öyle problemlerde de hakikaten onlar hani deep learning falandan daha iyi çalışıyor. Eğer veri küçükse, tabular dataysa vesaire efektif oluyor. Ama bu ilaç endüstrisindeki işte açık veri paylaşımı yaygın değil, evet, ama bu da değişmeye başladı. Geçenlerde denk geldim ben de. Melody diye bir proje yapmışlar. Federated Machine Learning yapıyorlar. Federated Machine Learning'de işte herkes kendi nodunda, kendi datasında computation yapıyor. Ama işte secure bir şekilde o prediction'lar birleştiriliyor şeyde. Yani kimse kimsenin datasını görmüyor ama aslında machine learning modeli onların her birinden aldığı bilgiyle daha iyi bir model train edebiliyor. Yani öyle şeylerin de önü açık bence. Test konusu düşünen varsa. Benim alanım değil ama.
Cevabınız için teşekkür ederim hocam. Bir sonraki sorumuz katılımcımızdan geliyor. CNN-based deep learning model is most suitable as an artificial intelligence classifier or hard sort, so it takes much time in the learning phase compared to machine learning. Can a low-cost heart disease screening system be developed using a CNN-based deep learning model and random forest classifier?
Evet, yani bu soru aslında şu, hani kalp datası, kalp ritim datasından CNN based bir deep learning modeli geliştirip hastalıkları yapabilir miyiz? Daha yani hatalı da olsa bir screening sistemi geliştirebiliriz. Bu da yapılıyor. Bu Andrew Ng'yi biliyorsunuzdur. Onların grubunda vardı bu kardiyografi datasıyla, nasıl söyleniyor şimdi bilemedim. İşte kalp ritmi verisini alıp orada da hani yani sinyali nasıl deep learning modeline sokarsanız işte CNN bunlardan bir yöntem. Öyle basılmış Nature'da bir paper'ları var. Nature Medicine'de sanırım. Çalışılıyor bunların üzerinde. Yani bunları ama tabii hani klinikte uygulanabilir seviyeye getirmek ilginç olacak şu anda. Research seviyesinde çoğu şey ama o kadar çok yatırım ve ilgi var ki machine learning'in sağlık alanına uygulamasına çok uzun sürmeyecek herhalde bu işler kullanılıyor olması.
Cevabınız için teşekkür ediyoruz hocam. Bir diğer soruya geçiyorum. Belirli bir modelin belirli bir etiketlenmemiş veri kümesi üzerinde doğru tahminler yapıp yapamayacağını tahmin etmek için mevcut bir doğrulama kümesi üzerinden neler yapılabilir?
Bir dakika, tekerleme gibi oldu, bir de anlamaya çalışayım. Bu arada yani sorular hem çok güzel hem de bayağı çok, biz hani seminer yapıyoruz. Böyle graduate öğrencilerimiz sağ olsunlar. Şimdi onları şikayet edeceğim. Graduate seminerleri yapıyoruz. 1-2 tane soru zorla geliyor burada. Bayağı siz beni sınava tabi tuttunuz. Belirli bir modelin belirli bir etiketlenmemiş veri kümesi üzerinde doğru tahminler yapıp yapamayacağını tahmin etmek için mevcut bir doğrulama kümesi üzerinde neler? Bu mi sorusu acaba? Out of distribution mı elimdeki veri? Yoksa benim modelimin train edildiği dataya yakın bir veri kümesi mi? Yani yanlış anlamadıysam bu da çalışılıyor. Yani benim eksperi olduğum bir alan değil ama bu yani ODD diyorlar işte out of distribution. Senin kendi train ettiğin verinin dışında bir özelliklere sahip bir veri kümesi geldiği zaman nasıl perform edecek? Bu out of distribution mudur, değil midir falan, bunların esas edilmesi. Var konuda da çalışma ama ben doğru insan değilim onu sormak için. Henüz oralara yetişemedim, bakamadım o literatüre.
Tamamdır hocam. Çok teşekkür ederim. Bir diğer soruya geçiyorum. Klinik uygulamalarda öngörülemeyen olumsuz etkilerin oluşturabileceği yan etkileri azaltmak için ne tür tahmin modelleri kullanılmaktadır? Stokastik süreçlerde bu modellerin güvenilirlik oranı nasıl bulunmaktadır?
Yani klinik uygulama derken bütün böyle o klinik uygulamaları mı? Bizim çalıştığımız bu arada benim bebek geliyor. 3 aylık bebeğim var da onu beslemem gerekecek birkaç dakikaya. Kapıdan ağlamaları gelmeye başladı sesi. Kusura bakmayın. Yani yan etki olarak biz işte drug çalıştık. Hani onunla ilgili bir şey söyleyebilirim. Orada değil, nedir onun adı, hani fena çalışmıyor ama yine de daha da iyileştirilebilir gibi. Mesela gene expression datasıyla biz datasının çok bir bilgi getirmediğini falan gördük. Ne öğrendiyse model drug structure datasından öğrendi. O niye? Onu hani o bir mystery aslında. Ya belki cell line expression datası tahmin ettiğimiz kadar kuvvetli değildi o problem için vesaire. Kapı çalıyorlar. Bir saniye geleceğim. Yani ama diğerleri için şimdi bir şey söyleyemeyeceğim. Yani diğer uygulamaların yan etkileri falan çok geniş bir alan. Hani hepsine de hakim değilim.
Tamam hocam. Cevabınız için çok teşekkür ederiz. Son olarak bizler gibi temel bilimlerde eğitim almakta olan öğrencilere akademi ve bilim yolunda neler önerirsiniz? Tavsiyeleriniz nelerdir?
Böyle araştırın diyeceğim. Biraz kuru alacak ama şu yaptıklarınız zaten yeterince ilginç bence. Hani bir sürü insanla konuşuyorsunuz. Eminim vizyonunuzu geliştirmek için başka şeyler de yapıyorsunuzdur. Okumak, staj yapmak. Yani farklı yerlerde. Yurt dışında da bir sürü olanaklar var. Onları böyle iyi takip etmek. Bazen bazı şeyler çok böyle hani şans eseri oluyor. Onu tanıyor falan. Nereden ne çıkacağı belli olmuyor. O yüzden bir de temelleri sağlam tutmak önemli bence temel bilimde. Kusura bakmayın, çok ağlıyor kapıda. Onu yapmam gerekecek. Bana yazın ama sonra eğer sorularınız olursa, olur mu?
Tamam hocam. Davetimizi kabul ettiğiniz ve çalışmalarınızı bizimle paylaştığınız için çok teşekkür ediyoruz hocam. Sizi aramızda görmekten büyük mutluluk duyduk.
Ben de öyle.
Sağlıklı günler dileriz.
Bu işi de tebrik ediyorum Bioinfocourse.
Çok teşekkür ediyoruz hocam.
Kolay gelsin. Görüşürüz.
Teşekkürler hocam. Değerli katılımcılarımız, Bioinfocourse kapsamında Haziran ayı etkinliklerimize katılım sağlayanlara Haziran ayı katılım belgesi verilecektir. 11 Haziran saat 8'de Sayın Profesör Doktor Mehmet Burçin Ünlü fizik ne işe yarar konulu bir seminer verecektir. Bioinfocourse platformu olarak etkinliklerimiz devam edecektir. Sosyal medya adreslerimizden takip edebilirsiniz. Hepinize katılımınız için teşekkür eder, iyi günler dileriz.